So just very shortly to introduce myself, I'm a senior researcher at Institute for Molecular Medicine Finland, and thanks to Brinton, who already gave from our sister lab from Sweden presentation of the basically basics what we are also doing for the precision medicine of solid tumors. So to the next slide. So basically, I'm now directly jumping to the phenotypic high content screening, but then actually describing you about a bit of our cancer project within the Finnish Academy of Finland ican flagship project, and also in the ERA-PERMED COMPASS project. We also nowadays screen quite often or do the phenotypic screening to the rare genetic diseases, plus then the drug testing based on the functional phenotypes we find. And we also work on the pathogens and more lately now for SARS-CoV-2, but this has been, of course, the kind of emerging COVID-19 and causing us a bit kind of repurpose our research partially towards that direction as well. But basically what we have developed are different types of the workflows for different type of the primary cells, followed up by high throughput screens, for example, such as described by Brinton on a drug testing site, high content microscopy, and then the machine learning based image analysis and some downstream analysis, as well as then kind of combination of the results of, for example, of the patient-derived cells from coming from other omics studies, transcriptomics, genomics, and so on in the molecular and clinical tumor part, to also present the results back in the precision medicine project back to the clinicians to actually improve the treatment of the patients. But some of these are more, as said, for the kind of basic biology, and some screens then are really like going towards the precision medicine. So this is the overview of ican flagship project. And here we basically link the tumor molecular profile data from the patients to their other type of digital health data, clinical data, and also longitudinal follow-up of thousands of patients. And now the prospective sample collection is over 5,000 to the end of 2022, and then 15,000 by the end of 2026. Of course, I mean, these samples are not all coming from our group. So we are actually here concentrating on starting from the patient in a couple of different types of the cancers, so urological cancers, and now also as a kind of very new project, also in the pediatric solid tumors. And the idea is that we really collect different type of the data, molecular data, and other type of the clinical data from these patients. And this data will be also later on available for everybody in kind of ican discovery platform for possibility to adaptive clinical trials, for example, for individualized therapies. But also then we have our own loop here in our research back to the returning this data back to the clinicians treating the patients. And this is example of our COMPORG project. It's about complex immune organoids for urological cancers, and we mainly concentrate on bladder cancer and renal cancer. And basically, after sampling of the blood and cancer tissue, in some cases also the benign tissue, we established the patient-derived 3D cultures, earlier mostly 2D cultures, but now we are concentrated more on the 3D cell cultures, as well as we do the PBMC isolation of these particular patients. We actually are now building up a complex immune organoid kind of co-cultures so that we could follow how, for example, the immune cells of the particular patient react together with the cancer cells from the tumor. We can also add their epithelial cells and fibroblasts, and this is now on what is ongoing. And of course, we work here also on the single cell sequencing of the particular tissue, and then of course of the immune organoids, as well as then very kind of how to say, deep characterization of these disease models, so that we can ensure that they are still representing the original tumor as much as we can achieve. Of course, we can't get back to the human physiology, but as close as we can get. And then here we also hope to develop advanced assays And this is now the collaboration with Vincenzo Cerula's group working on oncolytical adenoviral therapies to enhance the immune response of the tumors, for example, to the immune therapies, or for example, in T cells. And there are several different types of the approaches towards this direction. And this is one of them, which is more like collaboration for us, but I wanted to show it because I think that this is type of the new essays we kind of need to bring in. And here we actually have looked in renal and bladder cancer patient samples, organoids, so-called immunopeptidome, or have profiled it to actually enhance the T-cell mediated cancer killing. And basically, this type of knowledge on HLA +1 ligands from the tumor surface is actually quite important if we want to design these T-cell mediated cancer therapeutic approaches. And this peptide chip, which was designed by our collaborators, help us to identify and characterize these tumor-specific ligands. And basically, what we could actually then do was that when we found these ligands, we were able to then use the PBMCs, which were kind of pulsed with the identified peptides, so that we pulse them to be better effector cells. And we can actually see that there was the increased specific cytolysis of organoids by these T cells. So why do we do that? Of course, we want to kind of improve the therapies of the cancer, but then we even want to kind of enhance this immune response against tumor with this kind of peptide technology, but also we can then bring in also the modified viruses to further enhance this kind of peptide pulsed killing of the cancer cells. And here, what our PhD student Mikaela Fedorov has been working on is that she's been producing these different adenoviruses, which can actually then express or make the organoids to express chemokines when they are infected. And then there's a better tracking of these fluorescently labeled PBMCs, which, as I said, can be also then personalized way the peptide pulsed. So this is still in development, as I said, but we can already nicely see here the organoid. This is the renal cancer organoid prior to the virus infection, with expressing these cytokines, and then we can see how this organoid looks later when treated for 1 day with this oncolytic adenovirus. And we hope that we can now also develop this more towards a bit higher throughput format, these essays. Okay, and then I have no idea how much time I have because of some technical difficulties to start with. But the other project I just want to shortly describe you is COMPASS, which is the clinical implementation of multidimensional phenotypical drug sensitivities in pediatric precision oncology. And this is ERAPER-Med funded project, and why actually this was established was that together with 6 other European institutes, or actually one company or startup, we wanted to kind of find if we can use the standardized functional precision medicine setup to benefit the children with solid tumors, as they quite often harbor a very small amount of molecular alterations, and only some of those are actually druggable. So there's an urgent unmet medical need for these patients. And also the clinicians are very much into this type of approach. So basically, the aim I want to introduce here is to establishing a standardized ex vivo drug sensitivity testing platform, and the goal is to test 300 pediatric patient cases with relapsed tumors. So basically, this is also the U-bottom plate based setup, and we look here the 3D spheroids formed by these patient-derived cells. We usually do a very short propagation of the cells, less than 2 weeks, and we have also the COMPASS SOPs for both the cell culture for different type of tumors, as well as for the control cell lines, which we have been using for the standardization of this whole platform between different laboratories. And also at FIM, we actually print the COMPAS ready-to-go drug plates for all the labs on the regular basis. So then we have the drugs coming from the same source, and we are also controlling all the time how the performance of each drug. So there's a kind of very strict quality control for these samples. We work on the automated 3D image analysis. This is actually Peter Howard's team developing it. And then, of course, we want to integrate this data in the shared bioinformatics platform with other data from these patients. And actually, most of them come through other registry studies, such as INFORM study. And there's no direct clinical translation here, but anyway, the data is fed back via INFORM to those clinicians who send their patients to the INFORM study, and if the COMPAS screens are done also for those samples. Currently, there's about 100 patient samples being screened, and so far 80 with image-based readouts, mostly at Heidelberg by Sina Opperman and Ulo Witt and the team. Okay, and finally, I just jumped to the tools, which I think that we still need for this type of approaches. And of course, one of them is our kind of small toy, AI-guided automated spheroid picker, which we can use to select the spheroids of the certain size, for example, to establish high throughput screens where we have a very kind of regular size of all the spheroids or even organoids. And this was just actually set up to our institute this week, beginning of this week. So we are currently testing the system, and if you're interested, you can check the publication for it. And then at the end, also the other publication maybe of your interest would be the regression plane concept, which we just have published, and it's the analysis tool for detecting the cellular continuous transition states. So those biological processes which are basically continuous, and you can quite nicely there, for example, look certain processes such as mitosis. We also look at the blood cell differentiation and the how the lipid droplets are actually formulating in under different type of the treatments in the cells or perturbations. Okay, so to conclude and to finalize, this all needs and requires team science, and I think that it's great to be here today to discuss maybe how we all can develop this type of approaches together further. And at the end, want to just acknowledge all the collaborators and people involved in this study.